Dopamine, 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 dopamine. And don't forget norepinephrine, norepinephrine. Dopamine's best friend, norepinephrine.
Whoa there, that's not completely accurate. What about the cuddle hormone oxytocin?
Ooh, cuddling, yay!
Wait, are we talking about ligase? Yeah, I'm pretty sure that's what I did my research on.
No, Rob. Our protein is integrase. Where have you been?
What?
Integrase is much more important anyway. It's crucial for understanding important things like diseases.
You should be more specific. It plays a very important role in how HIV RNA gets incorporated into cells.
Ooh, that is more important. Retroviruses, man.
What's a retrovirus?
Say hello to my little friend RV, or retrovirus. He doesn't talk much.
He doesn't look friendly. I repeat, not a friendly.
He isn't. He rewrites your DNA through reverse transcription.
It messes up your life.
So, Integrase is a bad guy?
Because that's not what I want to study.
No, not really. He's just involved with bad guys. But for real though, integrase is super important.
Well, Corey, since you seem to have done your research on the right enzyme.
Rob, we all did the right research except you.
He at least tried to come prepared.
Why don't you explain it to me? I mean, us. The importance of the not-so-bad-guy integrase.
It was a dark and moonlit night.
When two dead boys got up to fight.
That's not accurate at all. Let's be serious.
Thanks, Shannon. Well, let's start with structure. Visual projections are great.
Poof!
Oh wow, it's a picture.
Yes, it's a convenient picture from prn.org.
It separates it so nicely. See, there's an N-terminus. It contains a C2H2 motif which binds to zinc.
Right, Rob. That formation allows the protein to maintain a folded structure needed for proper enzyme function. The zinc that is present allows it to stabilize its conformational changes. Let's eat the crust and then the middle of our protein. The C-terminal contains a SH3 fold.
SH3 is totes important. Doesn't it bind to DNA?
Yes, it does, but scientists don't exactly know how yet.
But what they do know is that integrase makes the process of binding the host cell's DNA possible, and how HIV-1 integrase gets, well, integrated.
You are so smart with your puns.
Scientists named it that way for some reason. I guess I just found it.
Oh, the cleverness of you.
So the last part of our segmented enzyme is a nice big middle. The core domain includes an RNA fold and is the site for all catalytic activity.
Side note, there's a line of positive amino acids which runs diagonally across the enzyme to stabilize the protein.
Ooh, look at it. It's so pretty.
Our drawing looks a bit confusing.
Voila! A better looking drawing.
Nice.
Well, I get the structure now, but why is it laid out that way?
The layout is due to its function and method of getting into the host cell. The enzyme itself serves two functions.
Ooh, and the first is to modify the 3' ends of the HIV DNA.
And the second is it acts as a helping verb.
In scientific English, please.
Sorry, it helps the transference of the viral DNA into the host DNA.
The HIV's DNA? So it's what aids the transferal of the viral DNA into the host cell's DNA? It doesn't originate in the host?
I got this one. Yes, HIV-1 integrase is responsible for not only modifying the viral DNA in the cytoplasm of the host cell, but it is also the enzyme that actually creates a 5 base pair break in the host DNA. Through that break, it inserts the viral DNA into the host genetic material in the nucleus.
Then once the ends are fused together,
The viral DNA becomes permanently associated with the host DNA to the point that the cell can no longer distinguish between the two.
Good job, Rob. You understand it.
However, integrase is not already present in the host cell. Integrase is created by a retrovirus, and in this case, that would be HIV.
Oh, I get it now.
Yeah, my good old friend RV is a father!
Baby, baby, oh,
So once the DNA of the viral HIV is inserted into the host cell, then it multiplies and spreads, which seems to be the point of any foreign or mutated thing. It has been the source of several superhero movies.
No need to get all philosophical about this.
Wait, you said integrase travels to the nucleus from the cytoplasm? That is such a long, slimy journey for a little guy.
Yeah, how does he do it?
Well, I don't know.
Scientists call it the pre-integration complex, or PIC, but that's just a label.
Yeah, it is something that is not really completely understood, yet it is still being studied. Wait, guys, it's being researched right now, as we speak. We should be more quiet. They need to focus.
It is really the perfect enzyme to study because it would allow scientists to specifically target HIV or other viruses that use integrase to get into the host DNA. This could lead to new treatment options that would allow good to conquer evil.
Sounds like biochemists have a lot of work to do.
It's so exciting and will be totally worth it.
I got this one. Yes.
You sure they are, Lindsay? Are you sure?
Yes, I got this right.